Good morning, sir. So, uh, I looked a little into the inner and outer mitochondrial membranes.
Okay, the question again, could you repeat just for everybody's?
The question was whether inner and outer mitochondrial membranes. Hold on, let me put on the correct video. Whether the inner and outer mitochondrial membranes are different or not, whether they have different compositions. And the quick short answer to that is yes, they have different compositions. More specifically, the inner membrane has much more protein content than than the outer membrane. And I suppose the primary reason for this is that the inner mitochondrial membrane has cristae foldings.
Okay. Sorry.
And those foldings house the electron transport chain. Sorry, where did I go?
No, you're fine now. Go ahead. Yeah. So you were talking about the cristae. What happens to the lipids?
1 minute, I couldn't hear you.
I was asking what happens to the lipid composition in the inner and the outer membrane. Is that
so? Yes, it does. Primarily, there are more lipids in the outer membrane than there are
more lipids. What does that mean? If you have a sheet of lipids,
so more lipids relative to the protein.
No, that's okay. So the question here is the composition, right? Protein content is another thing. As far as the lipid composition goes, is there a difference between the outer and the inner membrane? Tairia, can you hear me?
Composition is also different.
Okay. And are there lipids that are present in both?
Okay, now I
and
can.
Some lipids that are, different. Is that how it is?
So there are lipids that are present in the inner membrane, but not very concentrated in the outer membrane.
Sure.
One example is cardiolipin.
Okay.
So that is much more common in the inner membrane than it is in the outer membrane.
So is there a lipid in the inner membrane?
That,
can I ask you something? Is there a lipid in the inner membrane that is just not there in the outer membrane?
Yeah. Wait, I didn't catch you after inner membrane. I'm having.
Okay. So I was asking whether is there a lipid in the inner membrane that is completely missing in the outer membrane?
I don't think so. I don't recall reading about such a lipid.
Okay. So it's very likely that that there is a difference in composition, right? And that there are certain lipids that are enriched in one membrane as compared to the other, right? And it's possible that there is some kind of exchange that is happening, which means over a period of time, there isn't really a lipid that is uniquely present only in the inner, membrane, but not in the outer membrane, right? With proteins also, it's possible again that there are proteins that are enriched in one membrane versus the other. And, over time, there is possibility that there is some exchange that has taken place and you will see some of it in the outer membrane, but that there is so much in the inner membrane that probably this all, at some point of time could have been exclusively in the inner membrane as well, right? So that's probably how we want to think about this, right?
Okay. On that note, I read about there being contact sites where the inner and outer membrane come into contact.
Sure.
And there is a bit of diffusion of the lipids at those sites. So around those sites, we find equal compositions.
Sure. Sure.
in the outer.
So this is interesting also for other organelles when we think about it, that the membranes can actually be in contact with each other, in the sense that, for example, the ER and the Golgi will have contact sites. The ER and the mitochondria now are thought to have contact sites, and there are proteins that are actually present at these contact sites. So a lot of these organelles that we talk about now, that initially were thought to be very distinctly different structures, increasingly there is evidence to suggest that they all communicate with each other. Okay, so there is a possibility that things can go back and forth between them as well, in a way that we did not originally think of. Okay.
Right.
Okay, so thanks for that, Dhairya. Let's go to those who had questions last time. Let me see if I can find you here. Okay, Ajinkya had his hand up last time as well. Ajinkya, can you go first?
Yeah, so just one.
Sure. I can't hear you, Ajinkya. You're on mute.
Sir?
Yeah, now I can hear you.
Am I audible now?
Yes.
So I wanted to ask that yesterday you said cytoskeleton showed rapid reorganization. So can you specify in like time how quick they do it?
So this will vary, right? And these could be, the depolymerization can happen in a matter of seconds, milliseconds, right? And it also depends on many of these regulatory proteins that are present, right? So the presence of a regulatory protein can dramatically change the rate at which depolymerization or polymerization happens. It's a very rapid process. A lot of the cytoskeleton has been studied in vitro. So you can actually, put the components together and get them to polymerize, right? In Petri dishes, in slides, right? Or in cover slips. And the rate is very rapid, right? So it's seconds to milliseconds for like one subunit to be added, right? So it will take a couple of seconds for that strand to grow, and it can grow very rapidly. It can depolymerize very rapidly as well. Okay. Okay. Goraksh had put his hand up last time. You're around?
Yes, sir. Am I audible?
Yes, I can hear you. Go for it.
Is actin always a double helix, or it can be triple or multiples as well?
Yeah. No. So it almost always is a double stranded structure, right? So you won't get a single strand of actin. And it's a good question to think about, because this may have something to do with how it has stabilized, how these strands, are assembled. At some point of time, did a single strand exist before two were put together? We don't know. And it's interesting that even in the early prokaryotes, right, the MREB that we just saw is two strands wrapped on each other, right? Which says that right at the beginning, this may be vital for how these work. Okay.
Yes,
Chintamani Dhumal. Chintamani, can you ask?
Yes, sir.
Yes, go for it.
Am I audible?
Yes.
Sir, yesterday you said that about the introduction to the cytoskeleton, that cytoskeleton provides and encourages for an organisms. Could you explain in short about that?
Right. So they provide anchorage to organelles inside the cell, right? See, now that you know how the cytoskeleton looks, okay, imagine the cell filled with this cytoskeleton, right? The actin is running everywhere. The microtubules are running as long strands, across the cell. A lot of the the intracellular organelles actually, are needed to be kept in very specific places. Like the nucleus, for example, stays more or less in the center of the cell. In a migrating cell, the nucleus actually moves to the front of the cell. The Golgi, for example, needs to be right around where the empty OC is, right? The microtubule organizing center or the centrosome. So these organelles being kept in their place, they are for the most part bags of lipid that are floating around, right? But they have things on the surface. They have proteins on the surface. They have, motor proteins that could bind them as well. And so that allows them to be anchored and tied with the, actin or microtubule strands. And as a result, they are kept in place inside the cell, right? So the relative positioning of these organelles could be something that, is governed or controlled by the cytoskeleton. Okay, that's what I meant to say.
Yeah, yes, sir. And sir, one small doubt, sir. Yesterday you talk about some concept at redundancy. Is it okay to say as a supplementary, similar word to
So
redundancy?
supplementary is not redundancy, because supplementary could essentially say that what this does, this could come and help with. It could supplement that. That's not the case. In redundancy, this could do something. This could also do the same thing.
Yes.
Okay. And that's the difference in redundancy.
Okay, sir.
Okay.
Understood, sir. Thank you, sir.
Okay. Medha, you had a query?
Yes, sir. I wanted to know, like your slides had mentioned that microtubules help to determine the position of organelles in the cells.
Mm-hmm.
So I wanted to know, how exactly do they do that? Do they have is there a fixed template for the layout of the cell that they work off, or is it more of a continuous?
Good question. See, there is a kind of a loose template. It's not a fixed template. Okay. The organelles are also moving around inside the cell. And in response to certain stimuli, there might be very distinct changes in localization that happens. So remember, in a cell, the functionality of the cell is governed by these two parameters. It is governed by space and time. Okay. When I say space, it's what is happening where inside the cell. Okay. And time is what rate is it happening, right? When we talk about dynamics of actin cytoskeleton, the kinetics here, the rate is the important determining factor, right? Where the cytoskeletal polymerization is happening is another important factor. So every functionality in the cell is governed by this space and time, right? So the same is true for organelles. Their being kept in places is by the ability of the organelles to bind to many of the cytoskeletal components. Because the cytoskeleton is present everywhere, it can hold on to the cytoskeleton and try and keep its place, right? And that location may be determined by different cellular stimuli. So it's possible that, for example, when the cell has to move in a certain direction, the fact that the nucleus is repositioned to the front of the cell is a very elaborate mechanism, right? That allows the cell to do it. And that has benefits for the cell in terms of where the Golgi and the nucleus is relative to the front of the cell. So this is a dynamic thing. It's not like there is a direct template to say that if you look at it as a cell and you look at 37 degrees, you will see this organelle. No. But ballpark, you will know, for example, the nucleus is largely towards the center. The Golgi will be around the nucleus somewhere. So that relative positioning is maintained, right? And that positioning again contributes to this idea of space and time, where both these parameters are vital for any functionality to be regulated in cells. Okay.
I had a question regarding the microtubules that we talked about today.
Sure.
The polymerization and the depolymerization thing. Mm-hmm. So in case of when the cell is dividing, mm-hmm. Which side is the positive end and the negative end? If it attaches on both the sides, right? One is attached to
so the microtubules around the MTOC, the centrosome, is it positive end or minus end? Do you know?
I'm guessing it's the minus end. But
okay, look this up, right? That architecture is defined, right? See, remember, plus minus is also our terminology, right? What essentially you could call it, X and Y if you decide to, right? All it says is that the polarity is such that going this way, there is one kind of polarity. Going that way, there is a different kind of polarity. And if you are walking on the microtubule strand, you will feel it. You will feel what going this direction is like. You will feel what going in the other direction feels like, right? And you're right. The centrosome and the polarity of the microtubule that is attached to the centrosome is the same. So, this strand, if this is minus and it is plus at the other end, right? That end which is free is the one that is attached to chromosomes as it is being pulled towards the centrosome, right? So that architecture remains the same, right? It just uses that architecture differently.
No, my question was like, at the minus end, it starts depolymerizing, right?
Good question. Where does it depolymerize is the query.
No. Like, if it's attached at both the ends, then how does it depolymerize? Like, the depolymerization molecule should be in the centrosome or somewhere.
Right. So I'm going to send you something. As I said, the dynamic instability of microtubules is a very fascinating aspect of how polymerization happens, where it happens, where depolymerization happens, right? See, the critical thing is, this being a brief introduction to cells, I don't want to overload everybody with too much content.
Yes, sir. So my doubt was regarding that early prokaryotes didn't have any actin or motor proteins. Then how did they acquire the food?
So a lot of it was diffusion, right? They were, and actin and microtubules are not the determining factor for acquiring food, right? They are doing many more complex things. So obviously cells, survived in very primitive forms as well. Right? It's this complexity that's allowing them now to do many more intricate things. So, acquisition of food may not be stopped because they did not have motor proteins. Okay. Diffusion and other methods were available. It was not very efficient. It may not have been very controlled. All that is fine. But just if there was enough, to kind of to survive. And that's what they were trying to do.
Sir, I had a doubt, like in the last class, we studied that actin filaments and that microfilaments are same things.
So microfilaments can be actin, but there are, many specific proteins that also make microfilaments. Lamins, for example, keratin, for example, are very unique cytoskeletal components that make the intermediate filaments, right? In some cases, it's possible for actin to make thicker strands that resemble or make them resemble intermediate filaments. But actin is not intermediate filaments. Okay? Okay. Yes. Okay. Satvik.
Yeah, my question is the same. Like I had a question last time. It was about like what maintains the structure in the mitochondria. I looked some stuff up and I found things about mitochondrial structure, proteins, and the answer I got was that it's still being studied.
Yes. Yes.
So, like, is there are there microtubules inside the mitochondria? Like, are there
not in the form that that we know in the eukaryotic cell? Now, it's a good question. I don't know whether there is an FTSG like molecule in mitochondria. I actually don't know. Whether they have actually looked at something.
I read something about an experiment where they like modifications to the cytoskeleton affected the mitochondria structure.
That is possible, right? The modification of the cytoskeleton will affect the mitochondria. That doesn't mean the mitochondria itself has cytoskeleton.
Yeah.
Right. So this is that's a different question altogether.
Yeah. So
you're asking if the question is, what maintains the architecture of the mitochondria, there are, enough proteins that are playing an active role to do that. Does the mitochondria have its own cytoskeleton? I actually don't know whether that is indeed the case. Does the mitochondria talk to the cytoskeleton of the eukaryotic cell? Yes, it does. Right. So the location and even functionality seems to be affected by both actin and microtubules. But that's not because they are inside the mitochondria.
Yeah. Yeah. My doubt was just, is there a cytoskeleton like structure inside the mitochondria?
I don't know, actually. I actually don't know. Quick, Nilanshi.
Yes, sir. My question was regarding the regulator proteins that regulate the actin polymerization and depolymerization. The two that you mentioned right now, they were both either inhibiting the process of formation of actin filaments, or they were breaking up the already formed filaments. So are there proteins that support or promote this process of polymerization?
Yes, the short answer is yes. Right. And there are proteins that regulate the polymerization as well as the depolymerization. As I said, we just showed you a movie that kind of illustrates what's possible. There is a lot of information about regulatory proteins, entire reviews that have been written on just the proteins that regulate actin. And then similarly, an entire set of proteins that regulate microtubules. So it's actually remarkable that along with these structures existing in cells, the cells have invested a lot of resources in fine-tuning how these structures are assembled, disassembled, put together, because that's vital to cellular function.
Sir, my doubt was similar to microtubules have an anchorage at centromere from where they arise. Do actin also have something like that?
No, they don't actually. That's why, the idea of them being self-nucleating is very, very important for actin, because they have that ability that microtubules don't. Microtubules actually need a place from which to nucleate. So they can't just randomly start nucleating somewhere. Actin can. Okay.
So
and this is a big distinction in how they assemble.
I mean, it needs a support, right? So
its support is itself. Two such molecules can come together and two can become four. And before you know it, they are growing a strand.
Oh, okay. So self-nucleating. Okay.
It is self-nucleating. That's a big distinction. Okay. Okay. Good question. Good question. Arnav, final query.
Yes, sir. So you mentioned about how the microtubules present in the flagella, help in movement.
Right.
Yes. So my query was, what exactly controls the bending of the flagella? Is it the central microtubules or the peripheral?
So I think it's a combination, right? It's not just the center. Actually, the control happens because of the motor proteins walking. Okay, and the motor proteins actually bind the strands like this. So if these are the two motor proteins, this one is actually walking on this one is walking on this, right? And their movement, okay, is actually able to do this, okay, and allow the strand, the microtubule strands to move in this direction, which then when all of those strands do this, effectively translates into the movement of the cilia. Right. So that's how this process works, Arnav. Okay. Kedar, last question. You put your hand up late, very late. Okay. Very quick.
Sir, how the microtubules got direction to centriole to chromosome? How they got attached to centromere, not other else?
Okay. So that has to do with where they originate. Right. So they originate from the centromere. So it's not like microtubules were formed somewhere else and then found the centromere.
No,
they began at the centromere.
I am asking that how they are arising from centriole and they attach to centromere, no?
So you mean the kinetochore?
Yes, sir. Yes,
centromere is there, right? So kinetochore is at the chromosome.
Yes, sir. Attached to.
Yeah. So there, the same tip complex, right? I told you, remember that along with the originating, end of the microtubule, the tip of the microtubule is very vital for functionality, right? And that tip is actually what goes and anchors. So the tip has a tip complex, a whole bunch of proteins that are present there. Many of whom, will actually bind to proteins that are present here and on the chromosome, and then we'll be able to separate the chromosomes, during cell division. Okay.